Assalamu alaikum students. Today our topic of biochemistry is globular and fibrous proteins. So we have to learn about the globular and fibrous protein in this lecture. Before the start of the lecture, as you all know that there must be a certain learning objectives of that lecture. So at the end of the lecture, students will be able to identify the structural composition of proteins, understand the detail about the fibrous and globular proteins, and finally understand the difference between the fibrous and globular proteins. Before starting to our specific topic, fibrous and globular protein, we have to recall our previous knowledge regarding the proteins.
So, by definition, proteins are biopolymers. They are made up of different amino acids linked by the peptide bonds. So, when different amino acids they are joined by the peptide bond, they form the linear structure of protein molecule. That is a polypeptide chain. Actually, the polypeptide chain has a backbone that is a repeated sequence of NCC, where N is the N terminus or the amino terminus, and the C, that middle one C, that is the alpha carbon, and the last C, that is the C terminus, that is actually the carboxylic carbon. So this sequence is repeated again in the polypeptide backbone. In nature, all polypeptide chains are linear. They are not branched. When linear chains are overlapped over each other through the different bonds, they form the complex protein structure. Along with the amino group, alpha carbon, and the carboxylic carbon, protein also contains the side chain or a variable portion, or we can also call it the R group. That is actually not a part of the backbone of the peptide bond. But this represents the group attached with the protein molecules, and its importance is in the function and structure conformational of the proteins. So in this picture, you can easily see the different amino acids like amino acid number 1 and amino acid number 2. Both amino acids, as you have seen, that it has a purple color N group, the middle carbon, that is the alpha carbon, and the last carbon to which the two oxygen groups are attached, that is the carboxylic carbon. And another amino acid, that is the number 2, it again contains the same pattern of the sequencing. When two amino acids combine, they form the dipeptide. Actually, dipeptide, we call it the dipeptide because the bond that is formed is the peptide bond. It is a 1 in number, but two amino acids are formed. That's why it's known as the dipeptide. So the actual structure of the protein molecule which contains the amino acids is in a pattern of NCC. Again, when another amino acid is combined, it has a pattern of NCC.
Typical bonds that are present inside the protein molecule, they are the covalent bonds and the non-covalent bonds. Covalent bond includes the peptide bond, disulfide bonds, while the non-covalent bond includes the hydrogen bond, ionic bond and hydrophobic interaction. If there is a process of denaturation, so the process of denaturation occurs through certain agents. That process disturb or remove the all bonds from the protein molecule except the peptide bond. It means during the process and after the process of denaturation, all the bonds are disturbed while only the peptide bond remain intact. In this figure, as you have seen, that is the protein molecule. It contains the hydrogen bond, disulfide bridges, ionic bond and hydrophobic interaction along with the peptide bond. Peptide bond is not shown here, but you have to assume that a circular blue structures, they are the amino acid molecules, and peptide bond is present between the two amino acid forms the peptide bond. So the peptide bond is present between the individual amino acid. They are joined together to form the polypeptide chain. So by this structure, you can easily identify the location of different bonds that are present inside the protein molecule. After knowing about the different bonds that are present inside the protein to stabilize or to form the pure protein structure, so starting from the first, there is a formation of primary structure, then formation of secondary structure, tertiary and quaternary structure. These all structures are present inside the protein molecule. Now the question arises here, do all the protein contains these four structures? So the answer is yes, all protein contain at least the tertiary structure, while there are certain protein that contains the quaternary structure. So every protein molecule must have the primary, secondary and tertiary structure. So the native protein's structure is essential for the biological function of a protein molecule. Loss of a structure results in the loss of biological function. So in this picture, you clearly see the different structural composition of a protein molecule. Like primary structure is seen here, secondary structure, tertiary and quaternary structure. As you see in the primary structure, it is actually not a complex structure. It is a linear structure, linear chain of polypeptide. While secondary structure, as you have seen here, it is of two types, alpha helix structure and beta pleated sheets. Alpha helix is nothing but a spring like a shape of the protein molecule. It's just like a spring, and beta pleated sheets are just like a pleated sheets. Tertiary structure is actually the twisting and coiling of secondary structure over each other, while quaternary structure, as you have seen here, is actually the combination of the different tertiary structures. The first structure that has to be defined is the primary structure. Primary structure, what is it means? Actually, primary structure of protein is defined as the sequence of amino acids linked together to form the polypeptide chain. Each amino acid is linked to the next amino acid through the peptide bonds created during the protein biosynthesis process. So, it means that this structure that is formed by the combination of different amino acids, and you have seen that glycine is amino acid that is joined with the another amino acid, and the two amino acids are joined through the peptide bond. That is the strong bond that is not distorted or not destroyed. So the primary structure is formed after the process of translation. This is the initial chain that is formed in the process of protein synthesis. The primary structure, the peptides are formed. For example, dipeptides are formed, tripeptide, tetrapeptides are formed. Oligopeptide chain is also formed when the amino acid number are up to the 20. Polypeptides are formed when the amino acid ranges from the 20 to 50 amino acids in the chain, and whole the protein molecule is also formed when the more than 50 amino acids are present inside the chain. So this is a linear protein molecule, linear sequence of amino acid. It is a not complex process. It is a not complicated structure that is formed. It is a linear sequence of amino acid that is formed. Actually, it is a backbone of a protein molecule. Second structural organization is the secondary structure of protein. Actually, by definition, it is the regular local structure of the protein backbone stabilized by the intermolecular and sometimes intermolecular hydrogen bonds of the amide group. So, the special folding of a polypeptide chain in a properly arranged repetitive structure. This is known as the secondary structure of protein. There are three types of secondary structure of proteins, that are alpha helix, beta sheets, and beta turns. So the most common type of secondary structure are the alpha helix and beta pleated sheets. Both structures are held in a shape by the hydrogen bonds. We have read that the primary structure contains the peptide bond, while the secondary structures contain the hydrogen bond. Actually, the most important type of secondary structure of protein is the alpha helix. Especially alpha helix is present mostly in all the proteins. Alpha helix formed as a result of hydrogen bonds between the carbonyl oxygen and the amide hydrogen of the polypeptide chain and doesn't depend on the side radicals. The carbonyl oxygen of each peptide bond is linked to the hydrogen atom or hydrogen bond with the amide hydrogen of the fourth amino acid towards the C terminal. Secondary structures, especially the alpha helix that are formed early in protein folding, there are 15 to 16 foldings. They are stabilized by both sequence-dependent side chain of interaction and sequence-independent backbone interaction, especially the hydrogen bonds. Stretches or strands of proteins or peptides have a distinct characteristic local structure conformation or secondary structure depend on the hydrogen bonding. The two main types I have already described. The secondary structures are alpha helix and the beta plated sheets. Disulfide bridges are sometimes called the disulfide bonds or SS bond. They are actually the covalent link between the sulfur atom of two cysteine amino acids, and their formation stabilizes the tertiary and again higher order of protein structure. Special relationship of the different secondary structure, as you have seen, myoglobin actually is the structure of secondary structure, is the example of secondary structure, right? And lysozyme is also the enzyme that contains the secondary structure. Alpha amylase is an inhibitor, inhibitor of amylase enzyme that again contain the secondary structure. You have seen the three examples of secondary structure of protein. As you have looked, myoglobin actually made up of 75% of alpha helix. There are no beta pleated sheets or beta turns. It only contains the alpha helix 75%. Lysozyme contain about the 40% alpha helix and 12% beta sheets. So we have seen that it is not important that all the subtypes of secondary structure of protein are present in one molecule. Even one subdivision can be present in one protein molecule, just like a myoglobin only contains the alpha helical structure. And alpha amylase inhibitor, it only contains the beta sheets, not alpha spirals or alpha helix structure.
The third one is the tertiary structure of protein. Tertiary structure of protein is actually the complete three-dimensional shape of a given protein, 3D-like structure. It represents the special relationship of a different secondary structures to one another within a polypeptide chain, and how these secondary structures themselves fold into the three-dimensional pattern to form a protein molecule. Like in a figure, you have seen that brown or reddish bands are actually the alpha helical form, and the green lines there are shown by the arrow, these are the beta pleated sheets. So the combination of alpha and beta pleated sheets, repeated combination of alpha and beta pleated sheet, or the secondary structure of protein, the overlapping of repeated sequence of secondary structure of protein, then they form the 3D like structure, that is the tertiary structure of protein.
Okay, now about the last, quaternary structure of protein. Actually, quaternary structure of protein describes the joining of two or more polypeptide units. So when two or more, even four number of polypeptide chain or tertiary structure proteins are formed, they form the quaternary structure. The subunit Each have their own tertiary structure. Bonds which are present inside the quaternary structure, they are non-covalent interactions. So we have previously read that non-covalent interactions are ionic interactions. These are present inside the quaternary structure. Subunits can either functional independent or work cooperatively. So the different subunits that are present in one molecule of a quaternary structure of protein, they individually perform the different functions, or they even perform the functions combinedly. Dissociation of subunit results in the loss of the function. As you all know that if we dissociate the molecule of a protein, that it must lose its function. Now back to our topic, today's topic, that is on the fibrous and globular protein. So by the definition, the fibrous proteins are those proteins when the axial ratio of length to width of a protein molecule is more than 10. It is known as the fibrous protein. Examples of alpha keratin from hair and collagen, especially these two proteins are very much important as they come under the heading of fibrous protein. So you have to learn the definition or exact what are the fibrous protein. Actually fibrous protein, actually we have classified proteins into fibrous and globular on the basis of the structural organization. So when the axial ratio of length to width of a protein molecule is more than 10, then it is known as the fibrous protein. As in a turn, globular proteins are those proteins when the axial ratio of length to width is less than 10. It is known as the globular protein. Example of globular protein are myoglobin, hemoglobin, and ribonuclease, that is an enzyme present inside our body. These are the examples of globular protein.
The first example of globular protein is the myoglobin. What is myoglobin? Myoglobin is a protein that actually responsible for the storage of oxygen inside the muscles. Or it is a heme containing globular protein that is found in abundance in myocyte cells of heart and its skeletal muscles. Myoglobin contain actually the tertiary structure of protein that starts from the primary structure, secondary, and the tertiary structure. So in the myoglobin molecule, there is a repeatedly overlapping of especially the alpha helical structure. Oxygen binding pocket is formed by a heme group and a specific amino acid side chain that are brought into position by the tertiary structure. So myoglobin contain the tertiary structure of a protein molecule, and it includes inside the globular protein.
Another example of globular protein is very much important, that is the hemoglobin. A globular protein that consists of four subunits, 2 alpha and 2 beta of different types, right? Each subunit contains a heme group for oxygen binding. The binding of oxygen to one heme molecule facilitate oxygen binding by the other subunit. Replacement of even one amino acid in the primary structure with another amino acid, it is a critical for the function of protein, because if we replace one amino acid sequence or one amino acid place with the other amino acid, the whole function of the protein molecule is disturbed. Like that, as you have seen in that normal beta globulin chain in the hemoglobin, that is the DNA sequence, and that is the RNA sequence. As you already know that the DNA sequence is changed when it is complementary to the messenger RNA. It is changed. Like in DNA there is a C region, and when messenger RNA created, it replaces or it read or it binds with the G region, right? C always join with the G, T always thymine join with the adenine, cytosine again with the guanine. When this sequence is normally present inside the molecule, the amino acid that is made after the process of translation, glutamate is made, right? If there is a mutation in any gene, especially the gene that codes for the formation of glutamate, instead of this binding, as you have seen, there is a CAC code, and when messenger RNA read it, it forms the GUG code. The normal code was GAG. The abnormal code that is made is GUG because the DNA that is codon is altered. When alteration at genetic level, the messenger RNA got the wrong signals. So instead of forming the glutamate, it forms the valine. So when again this process continues, the abnormal amount and quality of polypeptide chain is formed. About the fibrous protein, fibrous proteins are much or most of the polypeptide chain in a parallel to the single axis. They have a single axis. Fibrous proteins are often mechanically strong and highly cross-linked. Fibrous proteins are usually insoluble in water, so usually play a structural role. The globular protein play a special role in the cell metabolism, while fibrous protein have a structural role. They have important role in formation of different structures of the body. So the important example of fibrous protein is a keratin, especially the alpha keratin. Alpha keratin are the fibrous protein. They make the hair, fur and nail tissues. Hair is made of twinned fibrils. Alpha keratin is rich in especially which type of protein? Cysteine, while beta keratin is deficient in it. So the differentiation, if anyone asks from you, the alpha keratin contains the cysteine and beta keratin doesn't contain the cysteine molecule. That is the alpha helix, coil of two alpha helixes. These are combining to form the two helical structures. First, the profilaments are made, pair of colloidal coils. Then the final filament of keratin is made. As you have seen that these colors shows the different coiling of a alpha helical structure of different types. Four right-handed twisted curves, protofibrils. That is the structure that is incorporated into skin, nails and furs. So the alpha helical structures are held together by the disulfide bond. Fibrous proteins that are again present inside our body, aside from the keratin, there are the fibrons, collagen and elastin. Fibrons are like a silk protein. They also form the spider webs. The spider web contain actually the fibrous protein which contains the fibrons. Collagen is an important fibrous protein. It is formed from the topocollagen subunits. The triple helix is topocollagen, is highly extended and strong. Again, it has a linear structure that is twisted and coiled. Elastin are the proteins present in the yellow elastic fibers of connective tissues, ligaments and tendons. So when we see at the different points, the fibrins, they are the silk proteins, the collagens are highly extended proteins, they are present in the collagen fibers. Proline is actually unusually high in the collagen fibers, and elastin are present in the ligaments, tendon and connective tissues. The differentiation points between the fibrous protein and the globular proteins, I have a big list of that, but we have to summarize it here in this slide. The first difference is that globular proteins are a compact protein structure. They have a compact structure, and fibrous proteins have extended structure. Globular proteins are soluble in water or in a lipid bilayer. Fibrous proteins insoluble in water or in lipid bilayer. The structural organization of the globular proteins, secondary structure is a complex with a mixture of alpha helix, beta sheets and loop structure. While in the fibrous protein, secondary structure is a simple with predominant one type only. Number fourth difference is the quaternary structure is held together by non-covalent forces in the globular protein, while the quaternary structure is usually held together by the covalent bonds in the fibrous protein, because they have a linear structure and they have a extended protein structure, so they have to be strong by the covalent interactions. As you all know that function in all aspects of the metabolism, just like enzyme, transport, immune protection, hormones, they are related with the globular protein. While the structural proteins that I have already told you, they are involved in the structural organization of different tissue formation, just like a tendon, bones, muscle, ligaments, hair and a skin. So that's all about today's topic. I sincerely appreciate your attention towards this lecture, and that brings us to the end. I would also thank you for your time and attention today. Thank you so much for your interest and attention. Jazakallah.